Okay, welcome to Heritage Classes. Myself, Usha, your botany teacher. So you had done your major test number 1. Topic was photosynthesis in the higher plants, respiration in plants, and then plant kingdom. I have put some entrance question majorly with respect to plant kingdom and rest, photosynthesis and respiration, some NCERT based questions. So let's see. Which of the following is not a product of the Krebs cycle? This is 91. So not a product, that will be NADPH2. ATP is formed, FADH2 is formed, and then NADH2 is formed. This NADPH2, this is not formed. So answer will be D. Okay. Okay, find the odd statement with respect to plant respiration. This is very simple. Glucose is derived from the end region of photosynthesis, that is sucrose, yes. As I here, see a simple concept here. In the case of plants, suppose this is a plant, this is a leaf here. Always remember that the product of photosynthesis is glucose. Okay, product of photosynthesis is glucose. But glucose, which is a product of photosynthesis, has to be converted into sucrose. Why? Because sucrose will be a transportable form or a mobile form. So first the glucose will be converted into sucrose, and then the sucrose will be loaded in the phloem. So it will be loaded from the leaf tissue to the phloem. So once it is loaded to the phloem, it will be in the form of sucrose. And then in the form of sucrose, it will be carried to different part of the plant, to the root, to the upper part of the plant, to every part of the plant. The transportable food will be in the form of sucrose. But remember that the product of photosynthesis was glucose. So glucose is converted into sucrose. Why? Because it has to be converted, it has to be transported or in the form of sucrose, because sucrose is a mobile form inside the phloem tissue. So when the plant cell will receive the nutrient inside the plant cell, it will not be in the form of glucose, it will be in the form of sucrose. So all the plant cells, they will receive nutrients in the form of sucrose. Now this sucrose, again, it has to break down to glucose and fructose. Then it has to enter the pathway of respiration. So in short, product of photosynthesis is glucose, but this glucose will be converted into sucrose, because sucrose is the mobile form. It will be loaded in the phloem and it will be transported to different part of the plant. Plant ka jo cells hota hai, they will receive nutrients only in the form of sucrose. So from the sucrose, it will break down to form glucose and then fructose. Then they will perform the respiration. So this is correct. Invert test, this is also correct. This is also correct. Third step, onwards are same for glucose and fructose. This is also absolutely correct. So the odd one in it will be this. Glucose and sucrose are phosphorylated in the presence of hexokinase. This is not related. Okay. Next, what is the speed of oxidation of NADH in case of fermentation? Fermentation means it will be in an anaerobic process. What will be the speed? The speed will be slow. But if it is aerobic respiration, then the speed will be vigorous. Okay. So it is asked here for fermentation, that means anaerobic respiration. So the speed will be slow. Next, the source of phosphorus in conversion of PgAl to 5-phosphoglyceric acid is. From where does this phosphorus comes in the conversion of phosphoglyceraldehyde, which is a triose phosphate, to 5-phosphoglyceric acid? That will be from inorganic phosphate. This is not given in the pathway properly in NCERT, but it is given in a written part. Like in the glycolysis pathway, from the PGL, it will convert to form 1,3-bi phosphoglyceric acid. Okay, 1,3-by-phosphoglyceric acid. In this 1,3-by-phosphoglyceric acid, this PGAL will undergo oxidation. So it will undergo oxidation, and that oxidation will release the electrons and the protons that will be accepted by NAD ion. Okay? That will be accepted by NAD ion, and it will convert to form NADH plus H or NADH2. So PGL will undergo oxidation and it will form NADH2. And it will also convert it to form 1,3-by-phosphoglyceric acid, and in it what is consumed here is phosphoric acid. And this phosphoric acid, phosphoglyceraldehyde, this will consist of one phosphate. Okay. So this phosphoglyceraldehyde will consist of one phosphate and 3 carbon. That is why it is called as a triose phosphate. So from one phosphate, it has now become 2 phosphate. By phosphoglycerate, 2 phosphate. The question is asking here, ये पहले तो phosphate एक था, तो दूसरा वाला phosphate जो है वो कहाँ से आया? That another phosphate comes from this phosphoric acid, which is from the external source. Okay, तो यहाँ से phosphoric acid से inorganic phosphate से ही add करके इस phosphate, यहाँ पे जो एक phosphate था, वो 2 phosphate बन के आ जाएगा by phosphoglyceric acid. तो इस स्टेप ऑफ ग्लाइकोलाइसिस में ऑक्सीडेशन भी होगा PGAL का, NAD आयन का रिडक्शन भी होगा, एंड इट ऑल्सो कंज्यूम्स दिस phosphate. Okay? So answer will be inorganic phosphate. In which of the following process carbon dioxide is not released? Carbon dioxide is not released in lactate fermentation. Next, during glycolysis, when glucose is catabolized to pyruvate, most of the energy of glucose is retained in the 2 molecules of pyruvate. Glucose was 6 carbon, and then in the glycolysis it has converted to form 2 pyruvate. So that 2 pyruvate will again enter the further process of aerobic respiration. Okay, so it is retained in the pyruvate. Next, oxidative phosphorylation yeast. What actually happens in oxidative phosphorylation? The reducing agent which are formed, that is the NADH2 and the FADH2, formed from glycolysis linking Krebs cycle. Those are oxidized in the process of ETS. I repeat, in the oxidative phosphorylation, the NADH2, FADH2, which are formed in the glycolysis linking and the Krebs cycle, are oxidized in the process of ETS. And because of the NADH2 and FADH2 oxidation, it creates a proton gradient. Where? It creates a proton gradient in the outer chamber of mitochondria. And that proton gradient, which has developed in the outer mitochondria, that proton gradient, when it breaks down by passing through F0, F1, at that time ATP is synthesized. So that energy of the electron, that means when the NADH2 and FADH2 were oxidized, in that oxidation, what happened? The electron used to pass in the electron transfer system. Because of that, it creates a proton gradient, and that proton gradient, when break down, it forms an ATP. Whole process is called as what? Oxidative phosphorylation. Because of NADH2 and FADH2 oxidize in the ETS, proton gradient develop, ATP is synthesized. So it should be formation of ATP by energy release from electron. Means energy release from electron means from the ETS process. Next, which of the following is an energy utilizing step in glycolysis? There are 2 energy consuming steps in the first half of glycolysis. That was the from glucose to glucose 6-phosphate and from fructose 6-phosphate to fructose 1/6 biphosphate. So answer will be fructose 6-phosphate to fructose 1/6 biphosphate, and it is intermediated by enzyme called as a phosphofructokinase, and this enzyme is called as a pacemaker enzyme. Pacemaker enzyme. It is a very important enzyme which will control the rate of glycolysis in the respiration process. See, inside the body, if agar already ATP high hai, okay, if there is already high ATP or if pyruvate amount is already high, then what happen? Or citric acid is already high. Citric acid high matlab Krebs cycle करने के लिए भी है. Pyruvate high means product of the glycolysis is high. So when already pyruvate is high, when already citric acid is high, when already ATP is high, जब ATP body में already है, तो ATP क्यों synthesize करना है ना? So that is why excess ATP and excess citric acid अगर है body में, so it will inhibit the activity of this enzyme, phosphofructokinase enzyme. I repeat, अगर बॉडी की सेल में ऑलरेडी एक्सेस एटीपी है या एक्सेस ऑलरेडी साइट्रिक एसिड है, देन फॉस्फोफ्रक्टोकाइनेज एंजाइम का जो एक्टिविटी जो है उसको इनहिबिट करेगा, क्योंकि ग्लाइकोलाइसिस प्रोसेस को स्लो डाउन करना पड़ेगा, क्रेब्स साइकिल को स्लो डाउन करना पड़ेगा. व्हाई? क्योंकि ऑलरेडी एटीपी एनर्जी है, देन उसको स्लो डाउन करना पड़ेगा. दैट इज व्हाई दिस फॉस्फोफ्रक्टोकाइनेज एक्टिविटी is inhibited by citric acid और excess ATP. बॉडी में ये रहने से इस एंजाइम का एक्टिविटी को इनहिबिट करेगा. दैट इज वाई दिस एंजाइम इज कॉल्ड एज अ पेसमेकर एंजाइम. एंड ये कौन सा स्टेप करता है? ये वाला स्टेप. ओके, नेक्स्ट, हाउ मेनी कार्बन डाइऑक्साइड आर रिमूव्ड फ्रॉम एसिटाइल कोएंजाइम A ड्यूरिंग क्रेब्स साइकिल? क्वेश्चन थोड़ा सा ट्विस्ट किया है, लेकिन मुझे आई फेल दिस क्वेश्चन वेरी नाइस. रिमूव फ्रॉम एसिटाइल कोएंजाइम ए मतलब एसिटाइल कोएंजाइम ए ही तो स्टार्ट करेंगे क्रेब्स साइकिल, है ना. एसिटाइल कोएंजाइम ए विल एंटर, इट विल बी एक्सेप्टेड बाय OAA, इट विल कंबाइन टू फॉर्म साइट्रिक एसिड, ओके, विच इज द फर्स्ट प्रोडक्ट ऑफ क्रेब्स साइकिल. एंड ये चलता रहेगा, क्रेब्स साइकिल पार्टवे चलता रहेगा. तो एसिटाइल कोएंजाइम जो था वो 2 कार्बन था. इट विल एंटर द क्रेब्स साइकिल एंड इट विल परफॉर्म द पार्टवे ऑफ द क्रेब्स साइकिल. यहाँ से क्वेश्चन पूछ रहा है कि हाउ मेनी कार्बन डाइऑक्साइड रिमूव फ्रॉम एसिटाइल कोएंजाइम ए ड्यूरिंग क्रेब्स साइकिल. क्रेब्स साइकिल के अंदर यहाँ से कितना कार्बन डाइऑक्साइड रिमूव होता है? व्हेन दिस विल एंटर, हाउ मेनी टाइम्स डीकार्बोक्सिलेशन ऑकर्स इनसाइड द क्रेब्स साइकिल? देयर विल बी 2 टाइम्स. तो आंसर विल बी 2, क्योंकि ये 1 मॉलिक्यूल का पूछा है, 1 एसिटाइल कोएंजाइम ए का. सो एक राउंड ऑफ क्रेब्स साइकिल में देयर विल बी 2 टाइम्स डीकार्बोक्सिलेशन. तो आंसर विल बी ए. नेक्स्ट, क्वेश्चन नंबर 100. All the following regarding the role of xanthophyll and carotenoid in photosynthesis are correct except. This will be except. यहाँ पे सिंपल सा बात है, their peak absorption is in the blue and the red region. ये blue and the red नहीं होगा, ये 400 से लेकर 500 के रेंज में रहता है. That is why it will be blue तो रहेगा but not red, it will be green. okay? इसका आंसर ये है. Another ये जो है फोटो ऑक्सीडेशन, ये क्या होता है? सपोज ये जितना ऑक्सीजन रिलीज होता है, द ऑक्सीजन रिलीज ड्यूरिंग द टाइम ऑफ फोटोलिसिस इन द फोटोसिंथेसिस, वो जो ऑक्सीजन रिलीज जो होता है जितना जल्दी हो सके क्लोरोप्लास्ट से बाहर, मतलब सेल में से बाहर जाना पड़ेगा टू द एनवायरनमेंट. अगर ऑक्सीजन एक्सेस रहेगा, देन दे विल ऑक्सीडाइज द क्लोरोफिल पिगमेंट एंड द अदर इलेक्ट्रॉन कैरियर. तो ऑक्सीजन ऑक्सीडाइजिंग द क्लोरोफिल पिगमेंट एंड द अदर इलेक्ट्रॉन कैरियर, उस फिनोमिनन को कहते हैं फोटो ऑक्सीडेशन. तो ये कौन प्रिवेंट करता है? कैरोटीनॉइड पिगमेंट. कैरोटीनॉइड पिगमेंट जो होता है, इट हैज़ अ स्ट्रॉन्ग एफिनिटी फॉर ऑक्सीजन. तो इसीलिए वो ऑक्सीजन को पूरा एब्जॉर्ब करता है एंड इट प्रिवेंट्स the photo-oxidation of chlorophyll molecule and other electron carriers. Okay. The primary role of oxygen in cellular respiration is to act as an acceptor for electron and hydrogen forming water. Last में ETS में electron किसके पास जाता है? To the oxygen, and that will be terminal electron acceptor, and उससे byproduct बनता है water. 102. During prolonged fasting, in what sequence are the following organic compounds used up by the body? Answer to this will be first carbohydrate, जो glycogen store रहता है, that will be first used. उसके बाद fat होगा, and at last that will be proteins. Next, what is the ATP utilized? What नहीं होना चाहिए, actually when होना चाहिए. When is the ATP utilized? ATP जो है किस फॉर्म पे यूटिलाइज होता है, कब होता है? ATP is utilized as and when needed. जब रिक्वायरमेंट है एंड जब भी रिक्वायर्ड होता है ATP जो है यूटिलाइज करेंगे. यही लाइन है, ये डायरेक्ट NCERT का लाइन है जो रेस्पिरेटरी बैलेंस शीट के पार्ट में दिया रहता है, एंड इट इज डायरेक्टली सेट कि ATP जो होता है इट इज यूटिलाइज एज एंड व्हेन नीडेड. रिक्वायरमेंट के अकॉर्डिंग यूटिलाइज होगा, लाइक दिस. 3A. 103. 104. The useful purpose served by lactate fermentation is. Lactate fermentation means anaerobic respiration का form है. What will it be? It will be regeneration of NAD ion. After that, consider the situation where photosynthesis is going on at steady pace. In a typical experiment with a light on and carbon dioxide is being combined with RUBP to produce phosphoglycerate. PG ही लिख दिया, phosphoglycerate मतलब PGA, phosphoglyceric acid वही है. Then suddenly the source of carbon dioxide is eliminated. कार्बन डाइऑक्साइड के सोर्स को eliminate किया. What will happen? प्रोडक्ट फॉर्म हो रहा था phosphoglycerate PGA and the acceptor is RUBP. अब कार्बन डाइऑक्साइड को रिमूव कर दिया, फिर क्या होगा? PGA मतलब phosphoglycerate का लेवल तो क्या हो जाएगा? प्रोडक्ट ऑफ द रिएक्शन वो तो क्या हो जाएगा? डाउन हो जाएगा. एंड जो एक्सेप्टर था RUBP केल्विन साइकिल का, कार्बन डाइऑक्साइड ही नहीं है तो RUBP एक्सेप्टर लेवल विल इंक्रीज. Just applied type का क्वेश्चन है. In which of the following reactions of glycolysis a molecule of water is removed from the substrate? ये जो होगा ये सेकेंड लास्ट स्टेप होगा. PGA to PEP एक स्टेप है. PEP to pyruvate ये स्टेप होता है. ग्लाइकोलाइसिस का 1, 2, 3 लास्ट 3 सबस्ट्रेट को लिख रही हूँ मैं यहाँ पे. तो PEP टू पायरुवेट जो होता है वो तो सबस्ट्रेट लेवल फॉस्फोरिलेशन होगा, ADP टू ATP, एंड PGA टू PEP होता है जो होता है द एंजाइम इज इनोलेस्ड एंड दिस इज अ डिहाइड्रेशन स्टेप, एंड यहाँ पे वाटर मॉलिक्यूल एक रिलीज होगा. ठीक है, तो आंसर विल बी PGA टू PGA टू, ये 2 PGA होगा, टू PEP. PGA to PEP. उसके बाद PEP to pyruvate. PEP to pyruvate to this will be substrate level phosphorylation. So 106 will be C. तो when you study, ये तो substrate level phosphorylation है. Glucose to glucose 6 phosphate ये तो energy consuming step है. Fructose 6 phosphate to fructose 1,6 biphosphate ये भी एनर्जी कंज्यूमिंग रिएक्शन है. तो एंजाइम एज वेल एज टाइप द रिएक्शन जो है वो बार बार इसमें लिख करके रखना सो दैट इट विल कीप ऑन रिवाइजिंग. तो आंसर तो सी हो गया. नेक्स्ट, विच ऑफ द फॉलोइंग स्टेटमेंट अबाउट फोटो रेस्पिरेशन इज करेक्ट? व्हाट हैपेंस इन फोटो रेस्पिरेशन? इट रिजल्ट्स इन द रिलीज ऑफ कार्बन डाइऑक्साइड एंड कंज्यूम्स ATP. ATP will be consumed but carbon dioxide will be released. Okay, this is 108. Correct option is, again a very simple question, gymnosperm will be seedrust. Seedrust, pinus, sequoia. These are all gymnosperms. The direct answer is this. And यहाँ पे wolffia, eucalyptus, this is angiosperm, and sequoia, the red wood tree, is a gymnosperm. Wolffia is the smallest angiospermic plant, and eucalyptus is the tallest angiospermic plant. Okay, is the tallest tree. Tallest gymnosperm जो है वो sequoia है, sequoia gigantea, and tallest angiosperm is eucalyptus. ये funaria, polytrichum is bryophyte, sphagnum is also bryophyte, polysiphonia is red algae. Equisetum is pteridophyte, adiantum is also pteridophyte, and the jingo is a gymnosperm. So answer is A. 109. Okay, how many oxygen are used and how many carbon dioxide are evolved? Answer to this will be this. Okay, 6 and 6. Yeah, लेकिन आया कैसे? In the case of respiration, glucose to ATP. In this step, first is glycolysis, second is Krebs cycle, third is ETS, fourth is oxidative phosphorylation. Okay, so in glycolysis, what happened? Glucose to pyruvate is formed, and pyruvate to acetyl coenzyme A is formed, and from acetyl coenzyme A it will perform the Krebs cycle, and from Krebs cycle it will have ETS and then to oxidative phosphorylation. ये है whole process of respiration. तो इसमें क्या होता है, यहाँ से यहाँ तक इस स्टेप में कार्बन डाइऑक्साइड रिलीज होता है, ठीक है, फिर acetyl coenzyme A will enter and it will perform a Krebs cycle. तो कितना कार्बन डाइऑक्साइड रिलीज होता है, 1 मॉलिक्यूल का 2 कार्बन डाइऑक्साइड, ठीक है. तो पाइरुवेट जब 1 मॉलिक्यूल होता है तो 1 कार्बन डाइऑक्साइड हियर, 2 कार्बन डाइऑक्साइड इफ इट इज 1 मॉलिक्यूल ऑफ एसिटाइल कोएंजाइम ए. ओके, तो 1 रोनो Krebs साइकिल का 2 कार्बन डाइऑक्साइड, पाइरुवेट टू एसिटाइल कोएंजाइम ए, यहाँ पे 1 कार्बन डाइऑक्साइड. तो कितना हो गया, दिस इज 1, दिस इज 2, 3. लेकिन ग्लाइकोलाइसिस से 1 पाइरुवेट होता है कि 2 पाइरुवेट होता है? ग्लाइकोलाइसिस से होता है 2 पाइरुवेट, यहाँ पे भी 2 पाइरुवेट. तो सब डबल हो जाएगा, ये 2 आ जाएगा और ये कितना आ जाएगा, 4 आ जाएगा, ठीक है. तो टोटल कार्बन डाइऑक्साइड कितना आ जाएगा, टोटल कार्बन डाइऑक्साइड विल बी इवॉल्व विल बी 6, ठीक है. यहाँ पे 2 कार्बन डाइऑक्साइड, ये डबल राउंड होगा, देन इट विल बी 4. एंड हाउ अबाउट द ऑक्सीजन? फिर ऑक्सीजन का कैसा होता है? ऑक्सीजन की केस में मुझे पूरा रब करना पड़ेगा. ऑक्सीजन की केस में ग्लूकोज टू एटीपी, अगेन रेस्पिरेशन प्रोसेस में 1 ग्लूकोज मॉलिक्यूल का 10 NADH2 and 2 FADH2 are formed. 10 NADH2 and 2 FADH2 are formed. That means total 12 होगा, ठीक है. तो 1 NADH2 का ऑक्सीडेशन में in the process of ETS it requires half oxygen molecule. 1 NADH2 का और 1 FADH2 का it requires half oxygen molecule. तो टोटल तो कितना हुआ, 12 हुआ ना. मतलब 1 NADH2 हो या 1 FADH2 हो, ऑक्सीडेशन में ETS में it consumes half oxygen molecule. तो टोटल तो 12 आ जाएगा ना. तो कितना ऑक्सीजन मॉलिक्यूल आ जाएगा, 6th ऑक्सीजन मॉलिक्यूल, ठीक है. तो कार्बन डाइऑक्साइड रिलीज विल बी 6th कार्बन डाइऑक्साइड, एंड ऑक्सीजन कंज्यूम जो है, ऑक्सीजन कंज्यूम will also be 6th. Okay. Next, that means answer is D. Next, the step of glycolysis where NADH is produced actually result in, ये वही है जो आगे मैं बताया था, उस स्टेप में PGAL का ऑक्सीडेशन होगा and it will form 1,3-5-phosphoglyceric acid. In this process, PGAL is oxidized. NAD ion will be reduced क्योंकि NAD ion जो एक्सटर्नल इलेक्ट्रॉन एक्सेप्टर है उसका रिडक्शन हो जाएगा एंड इसका रिडक्शन हो जाएगा. उसके बाद क्या प्रोड्यूस होता है 1,3-5-phosphoglyceric acid. And इस स्टेप में अगेन देयर इज यूज ऑफ इनऑर्गेनिक फॉस्फेट दैट मीन्स फॉस्फोरिक एसिड एंड प्रोडक्शन ऑफ, ये रिडक्शन करके क्या बनता है NADH प्लस ये बनता है. ओके, so it will be this. 10, 11. Energy releasing metabolic process in which substrate is oxidized without an external electron acceptor. That process will be called as an fermentation. Next, 12. Which statement correctly describe the light reaction of photosynthesis? What is the main aim of light reaction? That is production of ATP and NADPH2, which is called as an assimilatory power. 113. Which enzyme is responsible for the phosphorylation of ADP to ATP in mitochondria? That enzyme is very easy. It is called as ATP synthase. Crops such as tomatoes and bell pepper, ये C3 है, C3 plants है, allowed to grow in a carbon dioxide rich environment show higher yield. Why? Because they show an increased rate of photosynthesis at higher carbon dioxide concentration. जो ग्रीन हाउस होता है उस ग्रीन हाउस में लाइट का इंटेंसिटी को रिड्यूस करता है. ये एनसीईआरटी का ही एग्जांपल है, टोमेटोज एंड बेल पेपर. ओके, C3 प्लांट्स है. जो ग्रीन हाउस होता है उसमें प्लास्टिक से या टीन से सनलाइट के इंटेंसिटी को रिड्यूस करता है, ढक के रखेगा. बिकॉज प्लांट रिक्वायर्स ओनली 10% ऑफ द फुल सनलाइट. लाइट का सैचुरेशन पॉइंट ही 10% ऑफ फुल सनलाइट. तो लाइट का इंटेंसिटी को रिड्यूस करेगा, तो सब ढक के रखेगा. एंड अगर प्लास्टिक से साथ ट्रैप करेगा तो कार्बन डाइऑक्साइड का कंसंट्रेशन को इंक्रीज करेगा. बिकॉज ऑब्वियसली कम्पैक्ट रहेगा, देन कार्बन डाइऑक्साइड कंसंट्रेशन विल बी इंक्रीज. इस प्रोसेस को कहते हैं कार्बन डाइऑक्साइड फर्टिलाइजेशन इफेक्ट. तो कार्बन डाइऑक्साइड तो हाई हो जाएगा. तो फॉर C3 प्लांट्स their first carbon dioxide fixer is rubisco, which is a bifunctional enzyme. तो जिसका environment में ज़्यादा है concentration, rubisco will fix that. मतलब rubisco can fix carbon dioxide as well as oxygen. So when carbon dioxide concentration is allowed to increase, rubisco will only fix carbon dioxide. Okay? So answer will be this. तो उसका photosynthesis rate high हो जाएगा because carbon dioxide concentration को increase कर देगा. Identify the statement that is true for C4 plants but not for C3 plants. This will be initially fixed carbon dioxide mesophyll cell as 4 carbon long compound. C4 plants में यही तो होता है, mesophyll cell में carbon dioxide is fixed by peptidase enzyme and what is formed? OAA is formed. Okay, this will be A. 4 carbon long compound. In the absence of oxygen, the primary purpose of fermentation is to regenerate NAD ion from NADH, allowing glycolysis to continue. Okay. Which of the sequence, which one is the correct sequence occurring in glycolysis? The 117 glycolysis. This will be glucose 6-phosphate, phosphoglyceraldehyde, yes, 3-PGA, PEP, and then pyruvate. The answer will be directly B. ATP production. This is what I have said about phosphofructokinase enzyme. During cellular respiration is controlled by allosteric inhibition. The control is the allosteric inhibition by ATP and citric acid of the enzyme that facilitated the formation of. ये किस enzyme की बात कर रहे हैं? It is talking about phosphofructokinase. Okay, this enzyme, what does it do? It converts fructose 1,6 biphosphate from fructose 6-phosphate. So it forms formation of fructose 1,6 biphosphate from fructose 6-phosphate. This step is done by phosphofructokinase enzyme. Okay, 119. Which of them does not support the respiratory balance sheet assumption? These three will support the respiratory balance sheet assumption, and the last one, this does not support the respiratory balance sheet assumption. For this one to explain, it will take time. So those who want to know it, you come separately, I will explain separately, because to explain this, I have to say many things, which is not possible in this video. 120. Fermentation takes place under anaerobic condition in all prokaryote and unicellular eukaryote. In all prokaryote, fermentation, it is not possible in all prokaryote. Prokaryote can have aerobic as well as anaerobic. So in all is not possible. It can be in some. In yeast fermentation, pyruvate is converted to carbon dioxide is possible, ethanol is possible, but not water. Water is not formed. Water will be formed only in the aerobic respiration. So both statements are false. Okay, we are entering to plant kingdom phase now. Choose the correct option. In Oomasteus female, wrong option. In Oomasteus, female gamete is smaller and motile, while male gamete is larger and non-motile. This is given wrongly. This female gamete is smaller. This will be, of course, larger, and motile, larger, and this should be non-motile. And male gamete should be smaller and motile. Smaller and motile. That means this is wrong. The answer is A. 121A. 122. Cycas and adiantum. Cycas is gymnosperm. Adiantum is pteridophyte. It is a fern. Now, what do they resemble? They resemble each other in having motile sperm. Cycas has a top shape like this. They have a top shape multiflagellate. Largest male gamete. They have a top shape. They have a top shape multiflagellate. Okay, top shape multiflagellate. Largest male gamete. Male gamete. Cycas is actually the connecting link between pteridophyte and gymnosperms. Why? Because the gamete of cycas is motile, while the gamete of the other gymnosperms are non-motile. Okay. They are motile and they have many flagella. And at the same time, ferns gamete are also motile, and they have that twisted spiral shape, and they have many flagella. So answer is C. 23. Find the correct option with respect to Moses. This will be Archegonia produce many eggs, not many, it should be one egg. Archegonia are multiflagellated. This is wrong. They have dominant and independent, not sporophyte. This will be gametophyte. So answer to this will be this correct option. 124. Which of them shows isogamy with non-flagellated gamete? This will be Spirogera. In the case of Spirogera, the gametes are non-motile and are non-flagellated. Liverworts, mosses and ferns, gametophyte, are free living. This is correct. Liverwort, mosses and fern, gametophyte, are free living. Gymnosperm and some ferns are heterosporous. This is also correct. Sexual reproduction in Fucus, Volvox and Albugo. Fucus is brown, Volvox is green algae, and Albugo is Phycomystis fungi. So in all of them, they will have oogamous type. Oogamous means the female gamete will be larger and they'll be static. The male gamete will be smaller and they'll be motile. So this is correct. One answer will be A, B, C. Okay, A, B, C. And both Pinus and Mercantia are dioecious. Both Pinus and Mercantia are dioecious. It is wrong because Pinus is monoecious, Mercantia is dioecious. Answer will be A, B, C. Okay. Heterosporous. Common in all of the three, Funaria is bryophyte, Rhapteris is fern, Jingo is gymnosperm. So all of them, they will have the presence of archegonia. Bryophyte does not have vascular tissue. Gymnosperm, the male gametophyte is not independent. So answer will be A. 27, wrong one. वही question है. Find a wrong one directly here. Spirogera, motile gamete. This will be non-motile. One 28. All these three are correct. Has chlorophyll A, C, and fucoxanthin. Archegonia correct. Haploid endosperm is a typical feature of gymnosperm. Mucor is a fungi and has biflagellated zoospores here. This is wrong. Mucor, answer with this, mucor should be non-motile. 129. Mosses and lichen are the first organisms to colonize a bare rock. This is correct. In the weathering of rock, in the soil formation from rock to soil, first organism to come is mosses and lichen. Selaginella is heterosporous. It is not homo. Coralloid root is a negatively geotropic root in cycas, and VM stands for vesicular arbuscular mycorrhiza. Where is this type of mycorrhiza found? This is found in the case of Pinus. Coralloid root will be found in cycas, but this is not found in the cycas. Coralloid root will have blue-green algae, okay, and mycorrhiza will be having symbiotic association with fungus. So this is wrong. Main plant body in bryophyte is, that means this is wrong. This is wrong. Main plant body in bryophyte is gametophyte, whereas in pteridophyte is sporophyte. Pteridophyte is sporophyte. This is correct. Means A is correct, D is correct. In gymnosperm, male and a female gametophyte are present within sporangia located on sporophyte. This is also correct. So in the gymnosperms, the main plant body will be sporophyte, and in it, it will have a cone, and inside the cone, they will have a microsporangia and megasporangia. Wrongly match again. Answer to this will be A directly. Polysiphonia is a red algae. What is the similarity between red algae and a blue-green algae? Red algae and blue-green algae are photosynthetic and both have the capacity to change the coloration depending on the wavelength of light they absorb, and blue-green algae and the red algae again are totally non-motile. They don't have flagella. A. Which of them is a unicellular algae? Answer to this will be chlorella and spirulina. This is unicellular. Next, gametophyte is not an independent living generation in, not an independent free living generation in, that will be gymnosperm. Next, in Selaginella, the embryo develops into, Selaginella means pteridophyte, the embryo will develop into sporophyte. 134. Which of the algae is filamentous? Algae filamentous will be Spirogera and Eulatrix. This will be colonial. This will be unicellular. Fucus is multicellular. Macroscopic है. Colonial, again this will be unicellular. So Spirogera and Eulatrix are green algae and filamentous. 135. Incorrect about green algae. Green algae, the incorrect will be, this will be inner layer of cellulose and this will be outer. And photopigments are chlorophyll A and B. ये correct है. Asexual production in spores produced in juice sporangia flagellated होगा. Pyrenoid is also correct. Pyrenoid is a it is a structure which is present inside the chloroplast and is found mainly in the case of green algae. It has a proteinaceous core surrounded by the starch sheet. Okay, pyrenoid जो होता है अंदर में प्रोटीन रहेगा, it will be surrounded by the starch sheet and is a structure found inside the chloroplast. The function of pyrenoid is storage of starch and starch synthesis. Starch synthesize भी होगा, starch storage भी होगा. Where will it be found? Number one, it will be found in the case of green algae and lower organism like Euglena. Euglena is protista, photosynthetic protist. So photosynthetic protist Euglena also has pyrenoid. Okay, so thank you. हाँ, कितना है? इस बार ज़्यादा ही हो गया.